Good afternoon, my students. I welcome you all to this live Zoom class for tomorrow's 7th unit test. We have the reproduction organisms, one topic, another one human reproduction and reproductive health. So totally we have 3 lessons for tomorrow's NEET examination. I hope you people are sincerely preparing for your NEET examinations. So the date is also, tentative date is also announced. So you need a very sincere preparation for your success in the forthcoming NDA NEET examination. So today we're going to discuss the 2 topics. Okay, one is reproduction organism, another one is human reproduction. From birth to death, it is in the life cycle. From birth to death, there are different biological processes that takes place. So the reproduction is one among. Okay, reproduction is one of the important the life process. That is from birth to death. Okay, the various events takes place. So what is reproduction? The reproduction is a biological. Okay, it is a biological process in which the living organisms have the capability of producing their own offspring. Okay, own offsprings, their own descendants, own individuals. So it is a very, very important process of all living organisms. So by this process only, the organism is able to maintain the continuity of the races or continuity of their races. Right? So here in animals, the reproduction process are classified into 2 types. One is asexual reproduction, another one is sexual reproduction. Asexual reproduction and sexual reproduction. In the asexual reproduction, the 3 points you have to remember. The important 3 points you have to remember. 1, the asexual reproduction is a uniparental. The asexual reproduction is a uniparental. Second one, in a unisexually reproducing organism, especially in the case of fission, there is no mortality, so they are generally called immortal. The immortal. Third one, in a asexually reproducing organism, there is no variation. No variation, and they do not have the evolutionary. Okay, there is no any evolutionary significance, because in a asexual method of reproduction, the genetic material that is divided and the cytoplasmic components are divided. These are distributed to their descendants or to their young ones. So in a asexual method of reproduction, there is no utilization of more energy. But in the case of sexual reproduction, it is a biparental. But biparental is not necessary. You remember this in hermaphrodite animal. In hermaphrodite animal, single organism possesses both male and female reproductive structures. Right? That is a biparental, or you can say uniparental also, because in hermaphrodite animal single organisms that are able to produce both male as well as the female gametes. Here, sexual reproducing organisms, right? The somatoplasmic cell or the body cells that dies, but only the gametes are considered as the immortal. Gametes are considered as immortal, and the variation is there, and the variation leads to evolution. Variation leads to evolution. So, only there is a compared to asexual method of reproduction, sexual method of reproduction is more advantageous. So, new species of there is a plants and new characteristics will develop in the organism. So, when compared to these two, sexually reproducing organisms is more advantageous. So, here in asexual method of reproduction, the two more points that you have to remember. One is the process called karyokinesis. Karyokinesis is nothing but nuclear division. Nothing but nuclear division that occurs. So during either binary fission or multiple fission, the first event is the karyokinesis. Nuclear division that occurs first, and followed by is a cytokinesis. Okay, followed by the cytokinesis. So cytokinesis, it is a cytoplasmic division. Cytokinesis. Okay, it is a cytoplasmic division. Okay, cytoplasmic division. So most of these the events, there is a asexual method of reproduction occurs during a favorable condition, but in some organism to protect themselves from the adverse situation, especially in the extreme temperature and also extreme cold, the animal reproduces by the asexual, by forming this outer thick layer. So it occurs more, sometimes very unfavorable condition also. So this asexual reproduction, these are actually different types are there. Different types. The first one, that is the fission. First one is the fission. Okay, fission. In fission, first the karyokinesis as said earlier, and then cytokinesis, the cell division that takes place. In fission, the two types are there. One is binary fission. One is binary fission. Another one is multiple fission. Second one is the multiple fission. Binary fission and multiple fission. So two types are there. So mostly we see in lower organisms like bacteria and the archaea group of organisms, right? These binary fission, multiple fission, or this kind of the asexual method of reproduction that takes place. So in the case of binary fission, we have the binary fission that is divided into three types. The binary fission that is divided into three types. The three types of binary fissions are, there is three types of binary fission. One is irregular type of binary fission, transverse type of binary fission, and longitudinal. Okay, longitudinal type of binary fission. There are three types of binary fission which takes place in the organisms, right? So you see this, the first one, is a irregular type of binary fission. It's seen in the case of there is amoeba. In amoeba, this is in a freshwater animal. Here during the process of fission is a contractile vacuole. Okay, this is a contractile vacuole disappears. The contractile vacuole, okay, disappears, right? And here the nucleus, and here one more similar view that is not given here, nucleolus. The nucleolus and contractile vacuole disappears, right? And this chromosome or the nuclear material that elongated, and here in this the chromosome structure that is equally divided in the sense the nuclear material that multiplies and produce more copies of chromosomes during a favorable condition. Favorable condition in the sense when the organisms get more nutrients, and after the nuclear material, the composition is increased, the first nuclear division occurs. So in this diagram you can see here is the nucleus, and also it is forming the nuclear membrane as well as nucleolus, and this contractile vacuole is a contractile vacuole reappears. The contractile vacuole reappears, right? And later this is a cytoplasmic constriction that occurs, and then single amoeba now is divided into two daughter amoebae. So in this case, the division is actually irregular in the sense the division or the fission may occur at any plane or any angle. So this type of binary fission is seen in the case of amoeba. The second type of the fission that takes place in okay, so look at this diagram. The second type of fission that is transverse binary fission. The transverse binary fission that occurs in the case of the organism called paramecium. The example, paramecium. Okay, paramecium is also a protozoan animal. Paramecium also the protozoan animal, and this occurs in the case of planaria also. Okay, this occurs in the planaria. Planaria is a good example for regeneration process. So in the case of paramecium, you can see the two nucleus are present. Here one is the macronucleus, this one is the micronucleus. Two nucleus. This macronucleus that divide by amitotic method of cell division. In amitotic method, the nucleus is divide without undergoing any chromosomal structural changes, and the micronucleus that undergoes mitotic. So this one is amitotic is the direct division. It's called a direct division, right? And after that, when the nuclear material that is produced or developed, this through transfer axis, the paramecium divides, right? Paramecium divides. In this, both macro as well as micronucleus that is equally distributed to these two daughter paramecium. Both the paramecium receives equal amount of this macronucleus as well as micronucleus, and other structures like the hypostome and all these actually develop, and that become as the separate that lead a separate life. And the third type of that is longitudinal. Okay, longitudinal. That is a type of fission. This longitudinal fission occurs in the case of euglena. Or in general, in examination, they may ask in which organism the longitudinal binary fission takes place. They may give the examples like euglena, or sometimes in the option they may give flagellates. Okay, they may give flagellates. So the flagellate organism, because the flagella is the locomotory organ, the flagellate organisms, So normally this single flagella is present. Only single flagella is present. Here the chromosomal materials, this nucleus and also nuclear body, that is chromatic chromosome, everything is actually multiplies more during favorable condition. And exactly in the middle or in the longitudinal axis, the division furrow appears. When the division furrow appears, here actually only, the flagellum is there. Here in this region, the basal body is present, exactly in the middle basal body region. Then furrow or depression occurs in the longitudinal axis. When occurs here in this longitudinal axis, here you can see when after cytoplasmic division or cytokinesis occurs, two daughter euglena is formed. Two daughter euglena. Here you can see only one flagellum, but later another flagellum is also formed from the basal cell. Basal cell, the actually place where the flagellar structure or flagellum origin. So generally the protozoans are classified based on their locomotory organ. Based on the presence of the locomotory organ, these are classified into different types. So these are binary fission that takes place, right, during favorable conditions. During favorable conditions. And the second one, next one is the multiple fission. Second type is the multiple fission. Multiple fission that takes place, right, is also during favorable condition. Here when the organisms it is equally divides, instead they produce they forms an outer thick covering. So we see this. Okay, you see this is a 3-layered cyst wall is formed. This is a unique structure. The cyst wall with the 3 layer is formed only during the multiple fission. Only during multiple fission. Okay, the 3-layered cyst wall is formed. Right? Here the nucleus is present inside. This is a very thick as well as protective wall that is formed. Protective wall is formed. Inside the nucleus is present. The single nucleus now divided into many nucleus. And the 3 layer that actually gives protection. Right? Gives protection to the cytoplasm as well as the nucleus. And in the case of cyst formation, then another method is there in multiplication. So if the organism divide and produce more copies of daughter amoebae and released into the water medium, we call this the multiplication. In multiplication, there is another method is the young cyst formation, or that is called young cystment. This young cystment, when I mention the young cystment, okay, young cystment that occurs during unfavorable condition. But this 3 layer that gives complete protection, and within that here is the nucleus. This one is nucleus. Each nucleus is surrounded by many, this cytoplasm and also producing new layer or new membranes around it. This structure is called pseudopodiospores. It is called pseudopodiospores. The pseudopodiospores, right? It is well protected by the cyst wall during unfavorable condition, and when favorable condition returns, this wall ruptures. The wall breaks up, and more the daughter amoeba is released. That is, young amoeba is released. Now, young amoeba that leads to an independent life. So this is a unique. It is a type of binary, multiple fission. That is a young cyst formation or young cystment that is seen in the case of amoeba. So amoeba reproduces by is a binary fission, multiple fission, especially in multiple fission, right? By young cyst formation, and others reproduces by this method during unfavorable conditions, because the organism is living in fresh water. Suppose in an examination, in Neet, they ask, right, in freshwater sources, dry or become, it is disappeared, right? In this situation, whether the bacteria, the amoeba is able to live or survive in the ecosystem or not. So this organism is able to survive, and they will be there in the form of a system when the original environmental conditions retain, or during rainy season, the organisms get plenty of food. Then again, the system will rupture, and the organism, the cyst develop into new this amoeba. So this type of fission, this is the case of this protozoan organisms. So next one, the next one is called strobilation. Next one is called strobilation. Next is called strobilation. The strobilation type of asexual reproduction is in the case of Aurelia. Remember, this Aurelia is a kind of jellyfish. A kind of jellyfish. Okay, it belongs to Coelenterata group of, phylum Coelenterata. The strobilation here this actually reproduces, okay, by transverse. Okay, look at this. So you can see right. So this young strobila, this is a young strobila. This undergoes successive transverse, right? Transverse, that is the division or the divide into transfer axis. Each segment, they're developing into a small larvae. Each one is divided into larvae. The larvae is called the Ephyra larvae. The larvae is called Ephyra larvae. But when this produce and arranged on one by one like this, and after producing more number of individuals or more number of these larval form, they are still in contact. They are not detached. These are not detached, and these actually developing one by one. Here you can see this Ephyra budding. Budding is that is producing more copies, but they are dividing in transverse axis and producing more more individual larvae, and all the larvae that actually is still together for a long time, and later these actually live into the water medium and become as the free living, developing into free living Aurelia. Become as the free living Aurelia. So this strobilation is another type of the asexual method of reproduction seen in the lower organisms. And another one is called plasmotomy. Next one is called plasmotomy. The plasmotomy type of that is the asexual method of reproduction that is seen in the organisms like Opalina. Okay, the organisms like Opalina, and also the organisms like Pilomixa. It is called Pilomixa. Okay, Opalina and Pilomixa. So in this, this type of asexual method of reproduction, you can see this. Again, asexual method of reproduction. It is a Pilomixa. The cells, all the cells, you can see the diagram. The cells are again nuclear cells. In this, you can see multinucleated organism. This one is a multinucleated. This multinucleated structure, they are producing more multinucleated daughter cells. Right? Not a single. This is a single. This is a Pilomixa organism that has many nucleus. This each, organisms able to produce is a multinucleated daughter cell. It is a unique characteristics of this Pyromixa. That is example. And Opalina is also another animal. It is also example for this plasmotomy. Tomy means cut. Plasmo is nothing but the protoplasmic components. Generally, one cell has only one nucleus, but here there are many nucleus are present. It is a multinucleated structure. This multinucleated matured organism produce multinucleated many daughter individuals. Many multinucleated daughter individuals. That is another unique characteristics of this. Asexual method of reproduction. So next one is the budding. Next one is the budding. Okay, the bud is nothing but a small outgrowth. It is nothing but small outgrowth, right? That mostly develops from the matured part of the organism. Matured part of the organism. So the budding it is further classified into two types. One is called exogenous budding. It is called exogenous budding. Another one is called endogenous budding. Exogenous budding and endogenous budding. This exogenous budding that is commonly found in the case of hydra. Found in the case of hydra. Now, hydra is also a freshwater is a organism. Okay, belongs to coelentrates. Belong to coelentrates. Here in hydra, you can see two examples are given. That is in the case of both this Leucosolenia is a sponge, belongs to porifera, and budding that is in hydra, that is belongs to coelentrate organisms. Here in this case of hydra, the exogenous budding that occurs here, both in this Leucosolenia and hydra. Here in the basal region, the small outgrowths appears like this. Small outgrowths, the bud-like structure appears. And during the development, in the case of coelentrata or in the case of hydra, the body wall has, this hydra body wall has ectoderm, endoderm, and in between mesoglia. Mesoglia is a jelly-like material is present. This ectoderm, endoderm, mesoglia, that everything is actually formed here in the bud also. And in the center regions, middle region, there is a gastrovascular cavity. The cavity is called a gastrovascular cavity. This the gastrovascular cavity is also that extend up to this tip of that bud, and the tip there is a small tentacles that appears. The tentacles, right? The area is called hypostome area. Around the hypostome, the tentacle forms, and when the young one is fully mature, this bud detached after developing mouth and also tentacle, they detached and they lead into an independent life. So in both the cases, you can see in Leucosolenia as well as in there is a hydra. The young ones are daughter cells produced by the process of budding. Right? It is a example for exogenous type of this budding. So you must know all the examples. Okay, exogenous type, the examples you must know. When you come to this endogenous type, this is also called a gemule formation. Okay, it is called a gemule formation. The gemule formation, this occurs in mostly the example. Remember, this gemule formation occurs in freshwater sponges. Okay, mostly in freshwater sponges. Mostly only one, one or two species that occurs in marine species, but freshwater sponges that reproduces by this exogenous type of budding. And here You see the budding or gemule formation. The gemule formation here. Okay, this is a diagram. Look at this diagram. Here a thick membrane, thick outer membrane is formed. Here this is a very thick outer membrane, right? Here the outer membrane, and inside there is an inner membrane. This actually formed during. I said it is this kind of gemule formation in sponges that is mostly in spongilla. Example spongilla, that is the freshwater sponge. The nucleus which is present, the nucleus is completely covered by this very thick both outer as well as inner membrane. When the ecosystem is completely become dry, right? These membranes, both the inner as well as the outer membrane, gives complete protection. And you can see there is an opening called a micropyle at the free end. Inside the cells undergoes continuous the mitotic cell division, right? Mitotic cell division, and produce more the spore-like cells. The spore-like cells are called archaeocytes. The cells are called. This one is important. Remember this. Okay, these are important. The archaeocyte cells are produced during gemule formation in sponges, particularly in freshwater sponges, and when the favorable condition returns, again this micropyle region is open, and all the archaeocytes that is released into the water medium that is environment, and that is developing into a complete fully developed sponge, because the sponges get attached on the substrate. Get attached on the substrate. So there is gemule formation is a unique type of that is found only in the case of sponges. Unique type of asexual method of reproduction that is seen in the case of sponges. Remember this. Okay. And the next one is the fragmentation. Next one is the fragmentation. So the fragmentation, when an organism that is divided into many pieces, the animal, the body parts, okay, the body parts that is broken into many pieces. Each piece is able to develop a complete organism. This you can the process is also called the pedal laceration. This same in the case of sea anemone. Okay, sea anemone. Again, it is also a Coelenterata group of organism. And the body is actually broken into many bits or many pieces. Each piece is able to develop as a complete organism. And you can see in the case of tapeworm also, but in tapeworm only the mature proglotid that are detached from the organism, and each proglotid has both male and female reproductive organs, and they develop into the complete organisms, right? And the last one in this case, regeneration. Okay, regeneration is nothing but it is the ability of an organism to develop the last part. Okay, last part of the body. Remember this LOS, last part of the body. So, is a regeneration or the power of regeneration is more in the case of lower organism, less in the case of higher organism. You have to remember, in human being, the only organ which is able to regenerate is which organ? The only organ which is able to regenerate is liver. Only liver. Okay, so suppose if the part of the liver cells get damaged, the damaged part can be developed or regenerated. And other than this portion, other than this part, if any organ is lost, this organ developed only for the process we call this healing process. The process is called healing process. But only liver able to that is regenerate in human being. The regeneration is further divided into two types. Right? So the one type is seen mostly in the lower organisms, that is called a marked axis. Okay, it is found in the lower organisms called a marked axis. Marked axis type of regeneration that develops that is only in lower organism, and another one is called epimorphosis. Okay, and the marked axis, as said, the first type of marked axis. So if we cut the animal, or if the organism is broken in any angle, for example, there is a well developed in the case of planaria. Okay, planaria, and this developed in the case of hydra also. Suppose you take a planaria, the organism, the structure is like this. Okay, the planaria is like this, right? And if you cut the organism, right, as many number of species, either in the longitudinal axis or in the vertical axis, okay, or in the oblique or slanting. So if you cut any angle, any plant, and as many as number of pieces, all the trees have the capacity to develop as a complete the organisms. That is called a marked axis type of, this is a marked axis type of regeneration that is seen in the case of planaria. This is the case of planaria. And you can see this same type of this, right? Marked axis type that you can see in a hydra also. You look at this diagram. Look at this diagram. Okay, see this. Okay, now this is a regeneration process. That is in the case of hydra. Then the hydra that is broken, or this we can make into pieces the vertical angle. So the when it's broken like this, the anterior part is able to develop the posterior part, and the posterior part is able to develop the anterior part. So in this case, the power of regeneration, okay, the power of regeneration that is present in all portions of the body, both the anterior region as well as posterior regions. So here is also you remember this, okay, planaria also. So here is the head, this is a middle piece part, posterior region, okay, and each part able to develop as a complete this is a planaria. Or if you cut the animal in this longitudinal axis also, that are two equally there is a developed or equal size the planaria is developed. So that means every cell or every part of the the organisms have the capacity to produce, right, or develop into a complete animal. That is the power of regeneration more in the case of that is planaria as well as hydra. The planaria and hydra. And we come to the next one. We come to the next here epimorphosis. In the case of epimorphosis, okay, epimorphosis, there is two types of it. In epimorphosis, the two types are there. One is called reparative type. Okay, one is called reparative type. Another one is called restorative type. restorative type. In the case of reparative, only the animals or the organisms that develops the last part only, right? They develop into the last part, the part of the animal. So especially in the case of lizard, you can see the lizard that is last their tail. Only the last part that is reparative type. You can see the higher animals like human beings. Suppose any body part is lost or any tissue is getting damaged, any organ is lost, right? Only the organ or the tissue, or not organ, the tissue is only developed, but the organ will never develop. That is seen in the case of higher organisms like man, right? But a restorative type of epimorphosis, that is epimorphosis, that is seen in the case of, right, starfish, and also the tail, okay, tail of lizard. So when these organs partially or fully is lost, right? Partially or fully is lost, this organ will regenerate and develop into the original size. But here, the characteristics that is actually present in the some lower organism. But when the organism in the evolutionary process, if you see this, when it is become more advantageous, slowly the organism lost their regenerative power. So once again, you recall these points like fission, okay, in fission, binary fission, multiple fission, encystation, stabilization, plasmotomy, budding, fragmentation, and regeneration. In this part, you must know more examples. Okay, you must know the examples. Only few questions, 1 or 2 questions only is come from this portion, but you must give more importance to the examples, especially in this portion. And the sexual reproduction, we come to the another type of reproduction. There is the sexual reproduction. You know that the male and female organisms are there. The male and female organisms produce gametes. These 2 types are there. So 2 types. One is called conjugation. Another one is called syngamy. Conjugation is a very primitive type of sexual reproduction. Is seen in the case of organisms like paramecium. Example, paramecium. And this seen, okay, paramecium and bacteria. So the organisms are generally called conjugants. So in the case of paramecium, you see this is also called a slipper animal plus. Here we have we have this hypostome and the nucleus are present. Both the macro and the micro nucleus are there, and throughout the body they have cilia. They remember the structures. So the organism which comes together, okay, come closer, and the nuclear material through conjugation tube that are transferred from one paramecium to the another paramecium. So this is a very primitive type that is in the case of bacteria also. So the organisms are called conjugants. In bacteria, the one which donates the nuclear material is called F plus or male, considered as male. Another one which receives, that is called F minus or female. Very simple primitive method of the sexual reproduction in the case of lower organisms. In the case of syngamy, nothing but fused, the gametes are fused. So male and female organism produce the male female gamete, and also male gamete like the ova and the sperm or egg or sperm. These two fuse together to form zygote. The zygote develop into the complete organism. It undergoes the various developmental stages of developmental process. from zygote to the complete organism. So that we will see in the next part in human reproduction. So the zygote is restoring the diploid or 2n number of chromosomes. Over egg has n number of chromosomes. Sperm also has n number of chromosomes. The zygote is a 2n or diploid in nature that is formed by fusion of these two gametes. So that's why the process called syngamy. It's called a syngamy, fusion of two gametes. So here, the different types of syngamy is there, like the isogamy, anisogamy, like that. You go through the different types also. Generally, in higher organism or in sexually reproducing organism, the fertilization will be external or internal. Okay, or internal. External fertilization is seen in the case of aquatic animals like the amphibians. Okay, amphibians and the fish. Internal type of that is seen in the case of higher organisms. That is from reptiles to mammals. Reptiles to mammals. Okay, and here once the embryo is developed, the embryo is formed, the embryo undergoes. Okay, complete. There is a complete developmental process and become as the the fully developed organisms. Right, that we will see in the next part. And the next one is the parthenogenesis. Parthenogenesis is nothing but development of young ones without the act of fertilization. Without fertilization. And here the gametes are produced. The gametes may develop either into male or female, or either male or female in certain group of organisms. So based on that, here this is classified into three types. That is called arbinotoki. So the only males are produced by arbinotoki. That is the case of honeybees. So this one is the important one. Arbinotoki type of parthenogenesis, right? Which occurs in the honeybees. Only males are produced, but next one is the teletoki. That is the only females are produced. Right? Females are produced by this parthenogenesis process. Example: this one is seen in the case of some moth. Moths are like a butterfly. Right? Moths are like a butterfly. This is the teletoki. In some cases, the amphitoki type of is both either male or female. Okay, that is produced. So here you must know the examples. Okay, these examples important. These examples are important. Okay, so this amphitoki type of that occurs in the case of aphis. That occurs in aphis, or it is also called green fly. It is a pest. Green fly is a pest or aphis. In aphis, the either male or female organisms that is produced from unfertilized egg. And there is another classification. You go through this another type of classification of parthenogenesis. Here one is complete parthenogenesis. Another one is incomplete parthenogenesis. Complete parthenogenesis, incomplete parthenogenesis. Complete parthenogenesis. Here only females are there. Only females are present. Okay, males are absent. So females produce egg. They produce egg. The egg female haploid egg again developing into female. So there is no fusion of male and female gametes in the case of the complete parthenogenesis the organism that is seen in the case of rock lizard. This one is seen in the case of rock lizard. Okay, rock lizard or lacertilia. Okay, it occurs. It is seen in the case of rock lizard or lacertilia. So only females are present, males are absent. Incomplete type, and is seen in the case of honeybees, or generally it is present in the case of social organisms like honeybees. In honeybees, in the beehive you can see queen, okay, female. The other females are workers. Other female is queen is also female. Is the workers, right? Here these are developing from 2n number of diploid zygote, developing from fertilized egg. But the males are drones developing from unfertilized egg. So in examination, the males only males or only drones are produced by the parthenogenesis process, but the female, that is the queen and also other worker bees, that is developing by the normal sexual reproduction process. The sexual reproduction process, right? So here you remember this sexually reproduced organism. Remember that the gametes are produced, fusion of gametes, zygote formation, but only one special type of this sexual method of reproduction is seen, that is parthenogenesis. Here eggs are produced, but the eggs either may develop into male organism or female organisms, or both in some cases. Here also you have to remember the examples, right? So now I will give you some questions. I will give you some assignment question, and you look after this assignment question. Then we'll discuss the next human reproduction. Human reproduction. You take it's not a break actually, right? You check these questions, and then after we completing our discussion in this question, we'll continue the next part, right? The next one. Okay, go through the question, and after completion of the discussion from this one, we'll continue the human reproduction next part. So find out the answer. You find out the answer to this question. You read all these questions and find out the answer, and I will discuss all these questions and we'll move on to the next topic. Okay, students, have you seen these questions? Okay, first analyze this one and move on to the next topic. Okay, so first question is gemmules. Gemmules are okay, the gemmules are special reproductive structure of asexual reproduction that is present in the case of sponges. Okay, that is seen in sponges, gemmule formation. Next, development of pluripotent gamete, that is woven to form an organism without the act of fertilization, that is called parthenogenesis. So, implantation, gametogenesis are the part of the sexual method of reproduction. Natural parthenogenesis, that is natural parthenogenesis, that is seen in the case of honeybees. Natural parthenogenesis in the case of honeybees, not in housefly. Okay, and Drosophila, both male and female organisms are present. Binary fission, fourth one. Binary fission is a form of there is a asexual method of reproduction. So we can't say this one is a vegetative method. Vegetative method that is seen in the plants only. In the plants, that is a root, stem parts that develop in organisms. Organisms can show both binary fission and multiple fission. Amoeba shows both the binary fission as well as multiple fission. Look at the diagram. Of course, there is a binary fission. You can easily, tell what type of binary fission. Suppose if you ask, it is a longitudinal type. Okay, it is a longitudinal type of binary fission. That is seen in the case of euglena or flagellate organisms. And the term blastogenesis. Blastogenesis, not formation of blastula, not formation of blastocyst or blastosphere. All these three occurs during sexual method of reproduction. So the asexual method of reproduction that is seen in some organisms is called as the blastogenesis, or it is also called somatogenesis. It is also called somatogenesis. And identify the statement: amoeba is immortal because no death. The components or the substances are equally distributed. Complete parthenogenesis, that is seen in the case of complete parthenogenesis. Just now I explained Lacertilia. Lacertilia is example for complete parthenogenesis. Next one, the structure for asexual reproduction sponges, of course, gemmule formation. Immortal cell in higher organisms. Immortal cell is the germ cells. Remaining body cells, these are the body cells. They never develop in an organism, but a germ cells able to develop in a complete organism. Again, natural parthenogenesis seen. Okay, natural parthenogenesis. That is seen in the case of Apis. This is in the case of there is Apis. Apis indica is honeybee and Lacerta. In all organisms there is see natural parthenogenesis in the case of unequal binary fission. Unequal binary fission is in fungus. Okay, there is a Saccharomyces cerevisiae. Saccharomyces cerevisiae is fungi is unequal binary fission. Transverse binary fission. Transverse binary fission not in Hydra, that is in Euglena. Okay, transverse binary fission in Euglena. Gemmulation is in freshwater sponges. So here the option is given marine sponges also, but you have to select the freshwater sponge is the correct one. Isogamy means similar type of gametes are used. Example, monocystis. Monocystis. In remaining all three only asexual method of reproduction takes place. And asexual reproduction results in rapid increase in number, no genetic variability, no genetic variation is there, and the production of clones, all are there. Multiple fission occurs. Multiple fission that is occurs in the case of plasmodium. Multiple fission occurs in the case of Plasmodium. Plasmodium, it causes malaria. Hydra reproduces budding. Okay, this is the kind of asexual method of reproduction. The development of egg without act of fertilization, parthenogenesis. So here parthenogenesis is important one. Parthenogenesis type of there is sexual reproduction. Parthenogenesis is a type of parthenogenesis is a type of this asexual reproduction. Parthenogenesis is a type of asexual method of reproduction without fusion of gametes. In honeybee, the drones are produced from the unfertilized state. The asexual reproduction sometimes disadvantages. It prevents animals from moving around to find a mate. So there is no finding their partner or males, and no need of producing, wasting more energy. No need of more energy. And which are the following are concerned with the asexual reproduction? Bud formation. Plasmotomy. First generally first karyokinesis occurs. But in the case of plasmotomy, here only cytokinesis alone occurs. This only cytokinesis. There is no karyokinesis. And the two process only cytokinesis occurs, so that a more number of nucleus are present in this case. So you go through like this. The remaining questions from this part, and you get ready, prepare well to attend all the questions from this course. When you come to this next one, in this diagram you can see budding founding. So this next page is budding founding. Yeast, this is in the case of Hydra, this is the sponges, not in the amoeba, not in plant. So A, C, D, where A is the fourth option is the correct one. Fourth option is the correct one. And a haploid number of chromosomes are found in gametes. Haploid. Which of the following is immortal? We already discussed germ cell. Sequential and the regular conversion of zygote, right, into an organism. The whole process we call the embryogenesis. But I'll come to the next part. If female reproductive organs mature before male, that is called a protogyny. This is also an important one. If male organism matures first, that is called a protandric. Male matures first, protandric. Female develops first, that is called a protogyny. Development of female gamete to form organization without fertilization, parthenogenesis. Not bisexual, that is hermaphrodite animal. Earthworm is the hermaphrodite animal. Remaining male and female organisms differs. And approximate age of sexual maturity in boys and girls. In boys, there is approximately 14 to 16. Okay, the question is boys and girls. 14 to 16 boys, 11 to 14 girls. Estrous cycle is a characteristic of the mammal other than primates. It's also like a menstrual cycle. Gametogenesis in animals occurs in gonads. The act of childbirth is called parturition process. And lifespan is also another important question is there. A horse 60 is correct. Lion is also correct. Parrot is also correct. But the carp is fish. No, just 1 year. This estrous cycle occurs in the there is behavioral changes. This is not characterized by this menopause. There is no menopause in the case of estrous cycle. And listen, stay tuned, and we will go to the next lesson. Okay, next lesson there is human reproduction. Okay, human reproduction. So in human, know that the male and female organisms produce The gametes are produced in gonads. Okay, the male organism produce male gamete called sperm, and the female they produce female gamete called ovum or egg. This egg and sperm fuse together to form zygote. The zygote is the 2n number or diploid. Okay, so in human being there are 46 chromosomes are present. Ovum is only 23. Sperm is also 23. So, by sexual reproduction, the organism restoring their original 2n number of chromosomes. 2n number of chromosomes. So there are different stages are there in this in sexual reproduction case of human being. That is from gametogenesis. Okay, gametogenesis to it is a complete development of an organism. That is called till the process called parturition process. Till parturition, from gametogenesis, right, till parturition, there is a different events or different processes are there. Gametogenesis, two types are there. One is called spermatogenesis. Another one is oogenesis. Spermatogenesis and oogenesis. So the spermatogenesis and oogenesis is occurs the respective gonads. So in male, the reproductive organs you must remember, but just I will explain the testis. Here there is a male gonad testis, and the remaining the accessory glands and also the external genitalia that we go through that. If you take the testis, here it is located in a extra abdominal region. Extra abdominal location that actually located in the scrotum sac. You remember this? Actually, in scrotum sac, the temperature is nearly 2 to 2.5 degrees centigrade is lesser than the body temperature, because this the low temperature or low in the sense lesser than the body temperature that helps to maintain the normal production and the viability of the sperm. Suppose if the temperature in the scrotum is also as high as our body temperature, the sperm will die, because sperms are more temperature sensitive cells. They are more temperature sensitive cells, and this is actually located in the scrotal sac. Suppose if the gonads not descend into the scrotum, that leads to sterility. Okay, that leads to sterility. That is also remember this sterility. Okay, that is they may occur because of this non-descendant of the testis, right? Non-descendant of testis. Now, if you look at the structure of testis, the internal structure of the testis, here right, look at the diagram. Here the internal structure of the testis. Here this outer sac, the outer sac-like structure is called this one is the scrotum. This one is outer scrotum. And inside there are 202. This structure here nearly 200 to 250 lobules are present. 200 to 250 lobules are present. In each lobule, every lobule, there are 2 to 4 small seminiferous. Okay, seminiferous tubules. Okay, 2 to 4 seminiferous tubules are present. So from these seminiferous tubules, actually there are 3 different types of specialized cells are present in the seminiferous tubules, right? Inside the seminiferous tubules. So if you take the seminiferous tubule structure, here if you the single lobule is like this, right? Single lobule. Here the seminiferous tubules are present here like this. Seminiferous tubules, and inside the seminiferous tubules, here is a special type of cells are there. That is called germinal epithelium. Cells are called germinal epithelium, and in between the germinal epithelium, here, right, is another type of cell is also there. So this one is the germinal epithelium, and this one is called Sertoli cells, and between this membrane, here another type of cell is present. These cells are called Leydig cell. Leydig cell. So remember this, Leydig cells are not present inside the seminiferous tubules, right? And from this, the duct arises. The duct arises like this. The duct is called retitestis. The retitestis joins together here. The retitestis join together, and here it forms the structure. This one is called, right? Is vasa efferentia. This is called vasa efferentia. So first, testis, lobules, seminiferous tubules. This one is called retitestis, and retitestis gets into this one is called vasa efferentia. It's called vasa efferentia, and that leads into this green structure. You see the diagram. The whole structure, highly coiled portion, we call this is the epididymis. Epididymis. This epididymis that is divided into 3 portions. Epididymis that is divided into 3 parts or 3 portions. One is called caudal, head region is called caput. Head region is called caput epididymis. Right? The body of this epididymis is called corpus. And the tail region is called cauda. Okay, cauda epididymis, corpus epididymis, and caput epididymis. So these are called the storerooms of this cell. So immature gametes produced from the germinal epithelium that is actually transported through retitestis, and here is the vasa efferentia. Then stored here, and the complete maturation takes place here in epididymis. So epididymis is called storehouse of the sperm, and after that it is actually transported via this vasa deferens. So the sequence you must know. Vasa efferentia. This one is called vasa deferentia. So vasa deferentia from both the sides join together to form is a urethra, and in addition to that there are 3 glands, accessory glands. 3 accessory glands. They secrete their secretions like seminal, vesicle. Okay, 3 accessory glands: seminal, vesicle, prostate gland, and Cowper's gland. So they secrete their secretion, and these secretions contain fructose, calcium ion, and especially the secretion of Cowper's gland that produce an alkaline fluid. So only in alkaline fluid these male gametes become more functional, more active. So if it is in acidic condition, the male gamete will die. So these 3 secretions that is released gametes. It is these act as a medium for transportation of the gametes. So this is commonly called a semen. So in semen, this is the gametes that is transported through urethra, right, to exterior. So this is a structure you remember, and if you see this the process of the spermatogenesis. The process of spermatogenesis. So I will explain both spermatogenesis as well as oogenesis. That is simultaneously. You look at this. Okay, diagram. Here, the entrance point. Listen, in spermatogenesis. In spermatogenesis. The spermatogonia mother cell or sperm mother cell by the process of mitotic cell division. Actually, during gamete formation, both mitosis as well as meiotic cell division takes place. This is an important neat question. Both mitosis and meiotic cell division takes place both in oogenesis as well as in spermatogenesis. Okay, both oogenesis and spermatogenesis. And in the case of spermatogenesis, two steps are there, two events are there. One is for formation of the spermatid. Up to this is one process. Up to this, right? And second step called spermiogenesis. This spermiogenesis is absent, or spermiogenesis-like process is absent in the case of oogenesis. Okay, this is absent because ovum, the egg is a non-motile or movable. It's a non-movable, right? Here, one spermatogonia that actually produces by mitotic cell division, and the more number of cells are produced during the multiplication phase. This cell is called primary cell. So the important neat question. Both the spermatogonia and the primary oocyte, they have 2n number of chromosomes or 46 chromosomes in the case of human being. 46 chromosomes are present in the case of human being. And the cell, this primary spermatocyte that undergoes first meiotic cell division. First meiosis is the actual meiotic division, and secondary spermatocytes are produced. Secondary spermatocytes, they have only 23 or n number of chromosomes. 23 or n number of chromosomes. 23 or there is only n number of chromosomes are present. Okay, so only n number. And this secondary spermatocyte will undergo second meiotic cell. Second meiosis is mitotic in nature. Mitotic. This is another important question. Okay, first meiosis is meiotic in nature. That is called a reduction division. Second meiosis mitotic in nature, and after second meiosis, the 4 haploid spermatids are produced. How many? 4. So from one spermatocyte, 4 spermatids are produced. In examination, right? Sometimes they ask to produce 100 spermatid. 100 spermatid or 100 spermatid is produced from how many primary spermatocytes? So from how many spermatocytes? One that gives 4 spermatid. So that means from 25 primary spermatocytes, 100 spermatids are produced. Or from like that, from 100 primary spermatocytes, how many spermatids are formed? That is about 400 spermatids are formed. So like that, they will ask the question. So you understand it clearly and you answer it, right? And another step is the spermiogenesis. So this actually during this spermiogenesis process, there is this immovable, non-movable this spermatid that are developing into acrosome. Acrosome is developing from the Golgi, Golgi apparatus. Is developing from Golgi apparatus, and the nucleus is present here, and inside the cytoplasm is that is concentrated, and then from the head region there is a small tail and this mitochondria. Okay, and inside this mitochondria, the spiral, there is a mitochondria, and here is anterior as well as posterior region that the centrioles are present, and from this the long tail-like structure appears. So conversion of this spermatid into this active spermatozoa, the process is called spermiogenesis. So this is the maturation. During maturation phase, inactive spermatid is converted into an active spermatozoa and developing the head, and here small neck region, remaining body piece, and also long flagella or tail. So inactive or immovable spermatozoa is become as an active spermatid. The process called spermiogenesis. Remember that from one primary spermatozoid, 4 spermatozoa or 4 spermatids are produced. So this you can easily understand. Okay, when you come to the oogenesis, remember that egg or ova is developing in the ovary. Egg or ova is developing in the ovary. The structure I will tell you. And the oogonia cell. Oogonia. Actually, oogonia nearly 1 million oogonia are produced in the ovary of the female fetus. The ovary of the female fetus. So here is in fetal life, right? When the female is in the young stage or before birth, nearly about 1 million egg cells are mitotic cell division, right? There is a more number of primary oocytes are produced. The oocytes are present in the ovary of the female fetus. And this primary oocyte, right? The primary oocyte never produce, or oogonia cell never develop after birth. This is another important question. After birth, there is no formation of any more oogonia or primary oocyte. Right? Oogonial cells are produced only before, right? Only before the childbirth. So how many cells are there in the female fetus uterus? Ovary. That is about 1 million egg cells. And these egg cells undergoes the developmental process, so that it undergoes first division. Okay, there is a mitotic cell division, and this primary oocyte, when enter into mitotic division, the meiosis, right? That occurs. There is a reduction division, and the reduction division also first meiosis, and especially in the first prophase stage. The first prophase stage itself, the meiotic cell division stops. Right? Meiotic cell division completely stops at the first meiosis stage itself, before ovulation. And now this primary oocyte, right? This primary oocyte, remember this primary oocyte. It is for example, the egg cell is like this. The primary oocyte, these are surrounded by, the cells are called granulosa cells. This is called granulosa cells. When the granulosa cells are surrounding, they are called primary follicles. Okay. Now the primary follicles, it get mature, and the further layer of cells that is formed around it, and it leaving a space inside, that is called antrum cavity. And when the further the cells that is present around it, these cells are called secondary follicles. And these secondary follicles are supported by the different layers. The different layers are called theca layers. They are surrounded by the layers called theca layers. then theca layers are developed. That is called tertiary follicles. They are called tertiary follicles. Now the primary oocyte, it undergoes the three different stages: primary follicles, secondary follicles, and tertiary follicles. Later, they get matured, become as a Graafian follicle. Okay, they become as a Graafian follicle. Up to this Graafian follicle, the nucleus and also the cell which are present in the primary oocyte stage itself. It is in primary oocyte stage itself. So this primary oocyte, remember this primary oocyte, up to this primary oocyte, here the complete division is not occurs, complete division, and later the primary oocyte particularly in this after the different follicular stage that undergoes first meiotic division and produce one polar body and one secondary oocyte. So the secondary oocyte is haploid in nature. First polar body is also haploid in nature, but the cytoplasmic components are not distributed to the first polar body. Only one X receiving this because this secondary oocyte. So mostly this is actually released during up to this, right? This secondary oocyte is formed during up to what is it the maturation, sexually maturation stage, right? So during menarche, right? During menarche, during every cyclic event or cyclic process, only the secondary oocytes are released. Secondary egg. Secondary oocyte or secondary egg. This secondary egg, right, fully mature and undergoes the completion of the second meiosis only after fertilization. Only after fertilization. Okay, after fertilization. And if the fertilization not occurs, remember this, fertilization not occurs, the egg will die in the secondary stage itself. The egg will die in the secondary stage itself. After completion of this fertilization, right, this secondary oocyte is become as an egg, complete or fully developed egg, is also produced the second polar bodies. Here, the question is, how many polar bodies are produced during oogenesis? How many polar bodies are produced? If you look at the diagram, you can see only two, right? But in some book it is given uncertain. It is uncertain. But from birth or from the development of ovary till the ovum formation, if you analyze this, the first polar body also that undergoes the division and produce the two more polar bodies. So at the end of division, three polar bodies are produced. Three polar bodies and one oocyte. It is called one oocyte. The complete or fully developed oocyte is produced. Okay, so you remember this female as well as the male reproductive system. And then we come to that ovary. You see this okay, this is a female reproductive system. Okay, so this is a female reproductive system. This is an ovary where the eggs are produced, right? The egg actually released here in the abdominal cavity. The funnel shaped region is called the fimbriae. Actually, the whole structure is called fallopian tube. It's called a fallopian tube or oviduct. The oviduct has three regions. The anterior portion is called infundibulum. It's called the infundibulum. And this one is the ampulla region. And the connection, this portion, okay, it connects here. It's called isthmus. isthmus. Okay, so here the ovary, the egg that is released. Here fimbriae is the finger-like projection. The egg enters into this infundibulum and is passing through this cavity and reaching this ampulla region. So ampulla region is the place where the process of fertilization takes place. The process of fertilization occurs, and after fertilization, this is actually uterus. Uterus is called perimetrium. Perimetrium, and a thick muscular portion is called myometrium. Myometrium, and inner this area, okay, is called endometrium. So the endometrium only undergoes the periodic cyclic changes during menstrual cycle. These glands grow from the basal region, and if the egg is fertilized, suppose the egg is fertilized, the male gamete when it's released here is actually passing through this the uterus. The space. The fertilization occurs after fertilization. This egg, fertilized egg, this rolling over this endometrium and get attached here in this portion. So that is called a implantation process. The process called a implantation. So the implantation occurs, mostly after the 16 cell stage, and 2 layers are formed. One is a chloroplast and the living cells, and chloroplast actually helps to attach with this endometrial wall or endometrial gland. Myometrium is the muscular layer. Perimetrium is the outermost layer. This is a perimetrium. Okay, perimetrium is the outermost layer. Right? So once the ovum is getting fertilized, get attached, the process is called implantation process. The process is called implantation process. You see then another diagram. Here is a ovary. Okay, inside the ovary, how the follicles are actually produced. Okay, the follicles are produced, and here you can see is the blood vessels which enters here. These are the primary follicles. Okay, these are the primary follicles. The primary follicles only undergoes the different stages. Primary follicles, secondary follicles, like that. The undergoes primary, secondary, and also tertiary follicles. Finally, it becomes as the grafted follicles. So here the ovum is actually present. It is called a secondary ovum. Remember this, it's not a completely developed egg. Completely developed egg. This grafted follicle cells, the matured and also ruptured, the egg is released. This one is released, the egg is liberated. The process is called ovulation. The process is called ovulation. The ovulation occurs, exactly at the 14th day of menstruation, right? Day of menstruation, and after that, the remaining follicular cells get converted into the corpus luteum, right? It's a corpus luteum. This corpus luteum finally disintegrates and becomes a corpus albuminum, that is destroyed. This corpus luteum, suppose if the egg is fertilized, here the egg is fertilized, get implanted here, the corpus luteum produces a hormone called progesterone. It produces a hormone called progesterone. This progesterone is called pregnancy hormone. Progesterone is called pregnancy hormone. And when the progesterone is actually present here, the fetus grows and fully develops and forming the 4 layers. There is embryonic layers. The 4 embryonic layers are amnion, choria, allantois, and yolk cell. So 4 membranes, that is forming around the fetus. And from this membrane, the placenta. Okay, that appears, the placenta connected with the mother's endometrium. So the mother feeds not only for a the mother feed for a children or child also, for a child also. And the various hormones are produced. Actually, the process of gametogenesis is controlled by two important hormones. One is this is commonly called gonadotropins. One is FSH and also LH hormone. The FSH it stimulates both gonads and responsible for the spermatogenesis and oogenesis. Right? But LH is LH hormone that responsible to produce the androgenic hormone. Androgenic hormone, that is a testosterone. Androgenic hormones in males. Androgenic hormone in males. The same LH hormone, right, is also called, in male it is called ICSH. This hormone stimulates the type of cell called Leydig cell. So the Leydig cells produce androgenic hormone that is responsible for the secondary sexual characteristics. So here you must know the role of various hormones. Okay. So both FSH, LH, right, estrogen, progesterone, the role of all four hormones. Okay, role of all four hormones. Now, the embryo is fully developed inside the uterus. And when actually get attached, the different hormones are produced by the mother organisms like human chorionic gonadotropins. This is an important hormone, and also human placental hormones is also produced. These hormones, right, produced when the embryo is getting or implanted in the uterine endometrium. If you observe this development of an embryo, actually you know that the embryo develops or it takes about 280 days. 280 days or 40 weeks. Okay, 40 weeks. Okay, 40 weeks. And here are 9 months. In this, the heartbeat that appears in the first month, end of first month. Okay, and the first trimester, more or less, the embryo structure is fully developed. The embryo structure is human form is actually develops, and skin and other structure that develops in the second month. In the fifth month, the eyelid, okay, and also eyelash that separates. Okay, eyelashes separates, and there is a hair which hair develops on head, and also the other body parts that develops at the end of second trimester period. Now the embryo when it is fully developed at the third trimester, end of third trimester, the reflex, okay, the reflex that is generated, and the hormone called oxytocin is produced from the neurohypophysis, and this causing the there is a fetal ejection. Okay, fetal ejection reflex. Okay, fetal ejection reflex. So because of this fetal ejection reflex, and also the contraction of the uterus, the embryo is expelled out. So in this lesson, you concentrate from formation of the gametes till the development of the fetus. In addition to that, the mammary glands and also the complete cyclic events that takes place in the woman's reproductive tract, and also you have to we can't see. So the menstrual cycle also, exactly the 14th day is called LH surge. The 14th day is called LH surge. So exactly at the day of 14, the ovulation occurs. Ovum is released from the ovary. So it is a neat point of view to remember this. In this lesson, you go through all the questions and also the diagrams, because the diagrams are more important. Diagrams are more important because if you know the parts, then only you can write the sequential order. Even in male reproductive system, the female reproductive system, and you have to go through the order wise. Then only you can write the answer without any mistakes. Okay. Now we'll go to the some assignment question. Again, you take a few minutes. The time, and we'll discuss these questions. Okay. We'll discuss these questions. Okay. Take some time, 10 minutes, and we'll discuss these questions, and then you can prepare for your test. Okay. Look at these questions. Check the questions. Find out the answer. That I will explain the answer to. All these questions. Go through these questions, please. Okay, students, we go to the question. See the first one. The enzyme hyaluronidase is synthesized in Golgi bodies. Okay, the Golgi bodies of acrosomes. The Golgi bodies of acrosome. Okay, so only the acrosome region produces this enzyme called hyaluronidase, which helps to penetrate the sperm into the egg. Second one, the products of the first maturation division of primary spermatocyte in testis is known as the primary spermatozoid produces the secondary spermatozoid. Primary spermatozoid produces the secondary spermatozoid. Not spermatid. Okay, spermatid is the that is produced from the spermatozoid, secondary spermatozoid. Acrosome is found in sperm, not in ovum, not in zygote and embryo. The nucleus of sperm is located in the head region. The nucleus is located in the head region. Spermiogenesis is the transformation of, which I already told this also, spermatid into spermatozoa. Spermatogonia is not directly converted into, spermatozoa. That is, spermatogonia that produce spermatid is converted into spermatogonia. Head of epidermis, head region is called caput region. Tail region is called cauda epidermis. If the vascular difference of man is surgically removed, right? Vascular differential is removed, surgically removed. Actually, semen will be there, but there is no sperm because the vascular main function of the vascular differential is to bring the semen. Okay, that brings sperm. So semen will be there because semen is produced by the different glands like seminal vesicle, prostate gland, and Cowper's gland. Prostate gland and Cowper's gland. Okay. The number of spermatozoa produced single primary spermatozoid, 4 cells are produced. But from single primary oocyte, only 1 egg is produced. Only 1 egg. The number of chromosome in a primary spermatocyte, that is same as that of spermatogonium. Okay, same as that of spermatogonium. 10th one. How many secondary spermatocytes are produced from 400 spermatozoids? Okay, how many spermatocytes are produced from 400 sperma? How many secondary spermatozoids? Here, this also you have to consider secondary. Then 4 spermatozoa. So from 200. But suppose instead of secondary, if you ask primary, there are 400 cells are produced. 400. So there is the spermatozoans are produced. So secondary spermatozoid divides only one. That is second meiosis. So only 200 spermatozoans are produced. Mitochondrial sperm occurs in. Mitochondria is present in the middle piece. In head nucleus, head region, nucleus is present. Acrosome is also present in the head region. The duct which carries flow from testis to caput epidermis is called vasa efferentia. But from epidermis to the urethra is vasa deferentia. So here vasa efferentia carries the sperm from testis to the anterior part of the head. This epidermis is called caput region, caput epidermis. Fructose is present. Fructose that is produced in the seminal vesicle. The human males, the acidity in the urethra is neutralized by the secretion of Cowper's gland. Cowper's gland producing the alkaline fluid. Okay. So which of the following mammals produce alkaline mucus? That is Cowper's gland. This Cowper's gland. And next one, the seminal vesicle. The seminal plasma. The seminal plasma is produced by all these things: seminal vesicle, prostate gland, and also Cowper's gland. Produce the fluid, that is semen. Graphene follicles found in the graphene follicle ovary of mammal, not in the testis, not in the liver, not in the ovary of frog. Right? And the follicle that rupture at the time of ovulation and converted into corpus luteum. The corpus luteum produces the hormone called progesterone. It is progesterone. Graphene follicles located in the stroma of ovaries. Is present in the stroma of ovaries, not in the germinal epithelium. The main function of the corpus luteum, so it secretes progesterone. This is a pregnancy hormone. Right? In human, the fertilization of ovum takes place in ampulla region. The three portions are there, right? Not in the ovary. Cervix is a part of the uterus. The fertilization not occurs in uterus. It occurs only in the ampulla region. And three regions are there. So the one is ampulla, another one is infundibulum, third one is isthmus. The part of the fallopian is closer to the ovary. Closer to the ovary is the infundibulum. I will show the infundibulum. And graphene follicles contain graphene follicles. Graphene follicles contain both theca interna as well as theca externa layer. Only after the theca layers are formed around the secondary follicle, it becomes the tertiary and also graphene follicles. Discharge of ovum from the graphene follicle is called ovulation. It's called ovulation. Oogenesis is the process of formation of liquor. Abortion is the death of fetus. Fertilization is the fusion of both male and female gametes. The mammalian corpus luteum produce that I already discussed. Bartholin glands of mammalian female. This is actually present in the female reproductive system. This corresponding to the gland in the mammalian male is the Cowper's gland. The secretion of Cowper's gland and also Bartholin glands are more or less same. The Bartholin gland occurs in female only. Okay, in female, Bartholin gland it is the female. Okay, and female actually not in male. Here is also not male. In female, helping the vestibular lubrication. So third option is the correct for this question. Vestibular lubrication. Right? And question number 28. A gland in female corresponding to the prostate gland. Prostate gland. We know that only these three: Cowper's gland present in male, Bartholin in female, bulbourethral in male. Bulbourethral is also called the Cowper's. Remember this. But equal to this, corresponding to the prostate gland. Here the answer is none of this. So what is the correct answer for this? It is called a correct answer for this para-urethral gland is the correct answer. Okay, para-urethral gland is the correct answer. The clitoris, okay, clitoris of female is homologous to penis of male. Homologous to the penis of male. Okay, here think of. See, look at this. It is not analogous to the penis. Okay, it is not. It is a functional. This one is a functional and not overgrown. So first option is the correct for clitoris. So here in this part, remember this. The corresponding organ that you have to remember. Okay, male and the female corresponding organ. You remember this. It's another important. So one mark. One mark. One. Next question number 30. Question number 30. The germ cells, gametes, gonads, they actually produce both meiosis and the mitosis. This also will be explained during the gametogenesis process. Both meiosis as well as mitosis takes place. The correct sequence correct sequence of cell stages in spermatogenesis. So remember this first, not spermatocyte. First is spermatogonia. So these 1 and 3 is absent. That is wrong. And the second spermatocyte. Okay, this is also wrong. So spermatid is spermatogonia. So second option is the correct. Second is the correct answer. The sequence also you must know. So the chart thoroughly you learn. The genetic material of sperm is located in the head region. This is the head region. Middle piece. Here the middle piece. Mitochondria is this. Middle piece of mitochondria. The middle piece of mammalian stone contains centriole is also there. Okay, both centrioles and the mitochondria. Centrioles and mitochondria. The lytic enzyme produced by the sperm, hyaluronidase. This is a hyaluronidase. The acrosome is derived from Golgi bodies. So nuclear mitochondria is a spiral mitochondria. This also you must know. Is a spiral mitochondria. Of course, mitochondria is there that produce energy in the form of ATP. But we actually during spermiogenesis, it is get kind of rod-shaped mitochondria is converted into spiral-shaped mitochondria. And then next one, acrosome. Acrosome that is present in the head region. That not to find ovum, not to swim, and it helps to penetrate the membrane of an ovum. Because actually, ovum is surrounded by many layers. Is actually destroyed or lysed. Then the egg is the egg that is become developed. Okay, and becomes the fertilized one, zygote. And the next term, which statement is false? Which statement is false? See the cell division to form the blastocyst that begins after the fertilized egg implanted in the endometrium of the uterus. Cell division of the blastocyst begins after fertilized egg implanted is wrong. Before implantation, because the egg implanted after the 8 to 16 cell stage. So not after implantation, the division occurs is wrong. See the second one. The placenta contains cell derived from both the embryo and the mother. Is correct. Follicle stimulating hormone stimulated production of. This is also correct. Human male can continue to produce sperm throughout life is also correct. And that is not possible in the case of female. Under the menopause stage, the female not able to produce it. They lost the ability to produce it. Next one. If an adult human female took a drug that inhibited the release of LH, luteinizing hormone, which of the following is correct? So, following would not occur because the menstrual cycle will occur. Menstrual cycle will occur because that is done by the hormone called FSH. The LH actually prevents the ovulation. Remember this. Release of an ovum from mature follicle is prevented. So this one is the correct answer. Correct answer. LH prevents ovulation. LH prevents ovulation. If because of the person take drug that inhibits the release of LH, the LH is inhibited, ovulation is also stopped. Okay, so remaining correct. So here the statement would not occur. The negative statement is given. So second is the okay. Next one. Look at the diagram. Label the diagram. The outermost membrane Okay, there are three membranes are given. Oh, sorry, four membranes. If you look at this, the question extra embryonic membrane shown. The figure amnion. Amnion is an embryo. This one is an embryo. The embryo is surrounded by the first layer is called amnion. Is an amnion. And next one is given the allantois here in this diagram. Second one is given allantois. Third one is given chorion. Fourth one is yolk sac. Listen, from this abdominal regions here, this embryo, the embryonic membrane develops is called a yolk sac. The fourth one is yolk sac. Remember this. Fourth one is yolk sac. The first one is the amnion. Okay, and the next one, this one is the allantois. Okay, that is the allantois. Allantois. And the one, okay, this one is the chorion. Chorion. So from this you have to select the answer. First amnion. Okay. Second, first one is the amnion. Here the fourth one is yolk sac. Second one is the second one is the allantois. Okay, second one. This one is the allantois. This one. Third one. This one is the allantois. Okay. Allantois. This one is the chorion. So three chorion, one amnion, two allantois, three chorion, fourth yolk sac. So from this you have to select one allantois. First one is allantois. Okay. First one is amnion. Wait. This one is the amnion. This is the one. Second, yolk sac is the fourth one. Fourth one is the yolk sac. Second is the allantois. Third is the chorion. So 1, 4, 2, 3 is the correct answer. Right? So we select the fourth one. The correct sequence in the formation of spermatozoa is a spermatogonia. Okay. And primary spermatozoid, secondary spermatozoid, spermatozoa. Spermatozoa is formed by the process of spermiogenesis. Trace the sperm cell from the structure where it is produced till fertilization of egg. So you have to select the event 1 by 1. Event 1 by 1. What is the first process? That is developed in the seminiferous tubule. First. Okay, from seminiferous tubule, not vasodifferential. From seminiferous tubules, not vasodifferential, not tubules. So epididymis goes to epididymis. From epididymis, they reach in the vasa differentia. From vasa differentia to the male urethra. From male urethra, then to the female reproductive tract. Then uterus. And then finally, okay, when you are reaching this, okay, fertilization occurs here in the fallopian tube. So here the question is up to fallopian tube. Okay, or the fertilization occurs, that is that occurs in the fallopian tube. So up to fertilization that we have to select. So first produce the seminiferous tubule, second passing through the epididymis, then vasa differentia second, then into urethra third, and then enter into this vagina, and then to the uterus, then at the fallopian tube. So we go once again. This is the second option is the correct answer. Next one, How are mature human sperm and ova similar? They have approximately same size is wrong. They are different size. They are formed before birth is also wrong. Each have flagellum. No. In female gamete there is no flagellum. They are produced from puberty until death. No. From puberty till menopause in female, but from puberty till death in case of male. So this is also wrong. All four are wrong. They have both the same number of chromosomes. How many chromosomes are there? Number of chromosomes are 40. Sorry. Right? 23. The number of chromosomes are 23 each. Okay, only 23 chromosomes or haploid chromosomes are present. Okay, 23 chromosomes are present. And fertilization human used to take place in fallopian tube, especially in the amla region. Soon after the three germ layers are formed, the developing embryo, the process of organogenesis starts. The human brain is formed from. Human brain is formed from outer ectoderm. This is an organogenesis process. Actually, by the single layer blastula. Now getting the cells are like this. This get converted into three-layered structure. The three-layered embryo is like this. Three-layered embryo. And here endodome. Here endodome. And then the ectoderm is actually developing in the outer region. Ectoderm. And inside here in this region there is a mesoderm cell. And during this morphogenetic movement, the cells are arranged in a particular position, and from the ectoderm, this is an outer ectoderm, the brain, sensory organs that develops. From the mesoderm, bone, muscles, kidney, these organs developing from this area, and from here there is endodome, the intestine, and also there is a digestive glands are developed. So like the three single-layered embryo is getting converted into three-layered embryo. So from ectoderm, brain, sensory organs are developed. Right? So my dear students, I will stop at this level, and you go through more number of questions, and you get thorough in this portion. Before I will send you the assignment questions. Before you sleep, you complete all these assignment questions and get ready for the tomorrow's test. I wish you all the best. Do well in the examinations because we have very short time. Right? Thank you very much. We'll meet in the next class. Thank you very much.